Welcome everybody to this fourth video from the video tutorial series. Here we will look into some demo simulations which are available. If you have downloaded the source code, then you should see a subfolder that is named demo, and there are a number of examples there, and we will just have a look at some of these just to see how one can implement some interesting simulations and models, and to get some insights into how you can essentially learn from these previous examples and adapt this code for your own purposes. So here we are going to have a look at the so-called tumor concept demo. So this is a demo that simulates tumor growing in an environment of healthy cells. And this example has actually been implemented by my previous student, Jean de Montigny. And in order to simulate this model, we essentially can type BioDynaMo demo tumor concept. So here you see that this line here confirms that this tumor concept example has been created. And if we go inside tumor concept, and then we go into the source folder, we see here two files, tumorconcept.cc and tumorconcept.h. And again, as previously done, we can have a look now into the code of the .h file here, and you can see that here we have a slightly longer script than the ones that we saw before. Now, just to very briefly explain what's going on, I cannot go into much detail, but what essentially is being done here is we create our own custom cell class, which extends the cell class that already was implemented, that we have already used before. And we define that this cell actually, when it divides, what exactly is going on after division. So that's actually one of the big advantages of having this kind of object-oriented approach, because we can now have objects and we can define what's going on once these objects, for instance, proliferate and divide and give rise to new daughter objects. And in this case, we check that if the event that this object has undergone is a cell division event, then then the daughters will continue to be in a state where they can divide. Essentially, that's what is being said here, and this is also a method that has to be implemented to characterize the dynamics of the cell division. And then we have here the setters and getters. So these are methods that allow to set a certain a value and to read a certain value. In this case, it can set this can divide field, or otherwise it can read it. And the same is true for the color. So afterwards, when we visualize it, it would be, of course, nice to have colors, different colors for different cell types. And that's why we have here these getters and setters for the color. And then we have also essentially here a structure for the growth module. So as I explained in the previous video, we have these so-called biology modules which define what the cell elements can do. And here we define a new module called the growth module, and there essentially we again define what's going on within the growth module now. So this is the run method, which is executed at every time step, and here we check, for instance, if the diameter is smaller than 8, then it grows, and here we create some random movement, which is basically by restricted within the limits of -2 to 2. So these are the maximum values in each dimension. And then here we essentially implement the movement. So we update the position of the cell by adding this random movement. And remember, this only happens if the diameter is smaller than 8. If it is larger than 8, then with a certain probability the cell divides or it becomes inactive. So if this random variable here is smaller than 0.8, then the cell divides, and with 20% probability it becomes incapable of division. And then we here have a look at the simulate function, where we essentially create again the simulation object. We have here some parameters for the boundaries of the simulation. And now, importantly here, we create a for loop where we instantiate a certain number of cells, in this case 2,400 cells, and they are randomly distributed within a certain volume, and they have a diameter of 7.5, and then we also set the color depending on their y coordinate. So we essentially make a discrete coloring scheme where the coloring changes along the y coordinate, and then we add all those cells to the simulation, and then we create here one single cell, a cancer cell, which is essentially an object of the class that we defined before, this myCell class. And of course we need to say that at the beginning it can divide, otherwise it wouldn't be a cancer cell. And we add this growth module that we defined before and add it to the simulation, and then we simulate it. Now if we want to see how that plays out, we go as before inside the folder where this example is created, and then we type BioDynaMo build, now it takes a little bit longer than before because there is also more code, and then we type BioDynaMo run. Okay, finished successfully. And now we can have a look actually at the output. I did not go through it in detail, but essentially also here there is a bdm.toml file, and if we now use paraview to look at the simulation output, what we can do is we can here load state, and then we go to output, and then we have this .pvsm file which we can load, and now you see here a lot of cells, about 2,400, and you cannot see much more. So let us now change the coloring so that you can see what I meant before when I said that the coloring changes depending on the y-axis. So here you see that we have different colors, and this is the y-axis, as you can see here. Now still you cannot really see anything happening here, and the reason is that the cancer cell is inside this bunch of cells. So what we can do now is actually we can create a thresholding effect to the visualization. So we can here add this threshold, and then we here type in 7 for the minimum of the cell color value that we display. Because before, if you don't have this minimum, then we display all the cell colors, and now we just display the cell colors between 7 and 8, I suppose. And now you see that the visualization changed. And you can also see that now we can really see what the cancer cells are doing. Now obviously this is a very short simulation, so let us slightly increase the time from 50 to, let's say, 250. And now if we go again here and type BioDynaMo build, then BioDynaMo run. Okay, and now let me just restart, because otherwise I need to slightly adapt the visualization thresholding effect. So here we now load the state again, which is now updated, of course. And then we add here this thresholding effect. And then we run the simulation. So you can see here now there are many more cells than before, and that's why you can essentially see the impact of changing the duration of the simulation. So before we only had 2 cells, and now you see we have maybe 16 or 32 cells. Okay, I hope this makes sense. Feel free to play a bit around with this. Of course, this is very difficult to grasp everything in one brief sweep. So I would recommend that you have a look at the code and change some parameters, change some lines of code, and see how this influences the cancer growth.